Welcome back. Today we are diving into the chemical senses. Let's study exactly what makes us smell and taste.
In this lab, we will explore how our nervous system detects chemical stimuli. We'll start by breaking down the microscopic anatomy of the nasal cavity for smell, followed by the structure of taste buds on the tongue.
Let's break down the anatomy of olfaction, starting with the specialized tissues in the roof of the nasal cavity. On a diagram or a histology slide, we first identify the olfactory epithelium. This layer contains the olfactory receptor cells, which are specialized bipolar neurons equipped with cilia to detect odor molecules. Supporting these neurons are the sustentacular cells, alongside basal stem cells that continuously replace damaged receptors, and goblet cells that secrete a protective layer of mucus. Deep to the epithelium sits the lamina propria. Here, we look for olfactory, or Bowman's glands. Their ducts extend up through the epithelium to flush the cilia with fluid, dissolving new odorants. Moving upward, the axons of the olfactory receptor cells form the olfactory nerve, or cranial nerve 1. These nerve bundles pass through the tiny olfactory foramina of the cribriform plate, which is part of the ethmoid bone. Finally, these nerves synapse with tract neurons inside the olfactory bulb. From here, the sensory signals travel along the olfactory tract directly to the brain, bypassing the thalamus entirely.
Before we look at the anatomy, let's discuss gustatory discrimination. Human taste is categorized into 5 primary sensations. First is sweet, which is stimulated by sugars, alcohols, and some amino acids. Second is salty, triggered by metal ions like sodium. Third is sour, produced by the hydrogen ions in acids. Fourth is bitter, activated by alkaloids like caffeine or aspirin, which often serves as a protective warning against toxins. Finally, there is umami, a savory or meaty taste discovered by Japanese researchers, which is stimulated by amino acids like glutamate. When looking at a tongue model or a histology slide, the surface is covered in stratified squamous epithelium, which features distinct bumps called papillae. We need to identify 4 main types of papillae. First are the fungiform papillae, which are mushroom-shaped and scattered across the tongue. Second are the large circumvallate papillae, arranged in a V shape at the back. Third are the foliate papillae, which form parallel ridges on the sides of the tongue. All 3 of these house our sensory organs for taste. The fourth type are filiform papillae. These are sharp and structural, providing friction but containing no taste buds. Zooming into a histology view of a circumvallate, foliate, or fungiform papilla, we can identify individual taste buds. Chemicals in our food dissolve in saliva and enter through the taste pore to reach the gustatory receptor cells. Supporting these are basal cells, which act as stem cells to regularly replace worn-out receptors. Finally, taste sensations are carried to the brain by 3 distinct cranial nerves. Cranial nerve 7, the facial nerve, handles the anterior 2/3 of the tongue. Cranial nerve 9, the glossopharyngeal nerve, serves the posterior 1/3. And cranial nerve 10, the vagus nerve, transmits taste signals from the pharynx and epiglottis.
Finally, let's look at how taste and smell work together to create what we call flavor. While chewing, food molecules travel backward through the pharynx and climb up into the nasal cavity. This is called retronasal olfaction. The brain merges these chemical aromas with the raw taste data from our tongue inside the frontal lobe. This powerful combination is why food tastes completely tasteless when a stuffed nose blocks your sense of smell.
Thank you so much for watching. I hope you enjoyed learning about how we smell and taste. Stay tuned, and we'll see you in the next learning video.